Well, thank you, Maggie, and thank you to the organizers of this seminar and organizers of the Cover meeting. It's been a pleasure to be here, and it is a great pleasure for me to present some parts of the work that my group is doing at Northern Arizona University. Like eh, just in the sense of the type of topics that I want to cover with Maggie during this mentorship mentee association or interaction. I decided to focus my presentation today on on my work on soil trophic interactions across microbial domains. Let me just show you. So my laboratory is the laboratory of microbial trophic interactions. And you will see that my previous idea of why I'm only talking to soils today, it's coming from. In my laboratory, we study, the microbiome, from insects, animals, to the soil and the rhizosphere. That's what we do. So here I have two examples of the work that we are doing. One of these animal systems that I study in the laboratory is the most devastating coffee pest of insect. You may know it because it's here in Hawaii since 2010. This is Hypotenemus hampi. Back when I was at the Berkeley lab, one of my projects was to elucidate what was the importance of the beetle's microbiota in caffeine detoxification. And many years back, we demonstrated that the beetle was able to survive toxic levels of caffeine due to its associations with bacteria. So I'm just giving a brief view of what we do in that one because that's not going to be the focus of the talk. But I wanted to share that in the laboratory now, now that I can do other projects not totally related to bioenergy or energy in general. We are now working on understanding the transcriptomic responses of the host, the microbiome, and also playing with its microbial communities. We are working really hard on isolating bacteriophages that target specifically caffeine-degrading bacteria. So we want to remove them to then see how will the host respond, how will the community respond, with the idea of one day, trying to create a new strategy for pest management. But today, I want to tell you about my work with soils, again, as I mentioned before. This is a drawing that I made several years ago, trying to show our view of what microbial communities in soil are. In my laboratory, I try to instigate all my students to think of microbial communities as every microbe present in the soil. It will be the phages, some of them you can see them there. It will be, of course, fungi, super important. You have amazing experts here on it. I hope I will learn a lot from all of you and maybe collaborate even further. And we have the prokaryotes, could be bacteria and archaea. And of course, we have protists in there. And you can see there that I also like throw a nematode, even though, when you go to try to apply to certain agencies, they will tell you nothing larger than a protist. But I think nematodes, we all know, are super important as mediators of microbial communities in soil. So I put them there and we think about them. So we have a common theme here, which is multitrophic microbial interactions. And we apply this idea to any system that we study. Again, these are just some few examples. So when we talk about microbial trophic interactions, this is a little view of what we want to imagine. For example, we will have there plant biomass, represented there as cellulose or hemicellulose. You will have fungi. You will have bacteria that will work in the degradation of that plant biomass, releasing products that other organisms can consume and share with each other. Then, of course, we will have predatory bacteria, parasitic bacteria that, kill other microbes, and then release a lot of necromass into the environment and release CO2. And you have here the organisms that are not in my heart per se, but that I adore. I hope I don't have them in my heart. You have here the protists. Here I have the representation of an amoeba. And just a form of interaction between amoeba and protists will be, the consumption of the prey, getting into the phagolysosome. But then you will have other organisms that will defend themselves by producing toxins, producing different even antibiotics and chemicals. Sometimes this will affect other members of the community, right? But then those that are not so lucky to have these weapons to protect themselves will be consumed. And through the consumption of these organisms, we will have the release of ammonia in different forms that can be consumed by organisms. And this will create a general loop of microbial associations. So in my group, we are trying, okay? We are working really hard to try to put together this picture in any ways that we can. One way in which I started doing this was back in the Berkeley lab, the last few years that I spent at the Lawrence Berkeley National Laboratory, was to use a natural experiment to ask how microbial communities reorganize as soils age and nutrients decline. This photo that I'm showing you here is my perception of the system that I was using, or that I keep using actually, located in Mendocino, California. This is the ecological staircase and is a retrogressive ecosystem. A retrogressive ecosystem is an ecosystem in which time and not a major disturbance like a fire or some destruction or anthropogenic inputs creating disturbances. Here, time is the main driver of changes in the ecosystem. You will have nutrient limitation and, of course, soil degradation. So that is a retrogressive ecosystem. What you can see in that drawing here, let me try is, well, my system of study. As I mentioned, we have limiting nutrients will be nitrogen and phosphorus. This has been demonstrated because there are many studies done in this ecological staircase. We have different terraces. My study focuses specifically on the young terrace that is known to be 100,000 years old. Then is the third terrace, which is 300,000 years old and 400,000 years old. I will call this one young, mid-age, and old. Regarding vegetation, which is a super important driver of community associations, in the young terrace, we have orchids, grasses, but we also have different pine trees. These same pine trees will also be very dominant in the mid-age and older terraces. They are present there. In older terraces, we also have a very important group of plants, erigot shrubs. I'm showing you the ones that are reported in these ecosystems. And this is the cool thing. This is a small terrace. It's like 5 miles, long. The elevation of the changes between terraces is only 2 meters when you're from young to old terrace. So there are no major environmental changes. There are climatic changes. One thing that I want to mention here is that if you go to that terrace, you have many of those here, but that's different. Okay, the one we have there is very different. You will see the tall pine trees that are present in terrace 1, terrace 2 that I didn't label here, are also present in terrace 3 and terrace 4, but they are in dwarf form. It's the same species, but they have adapted to actually preserve nutrients and they do not grow like the others. They are simply retaining nutrients and try to keep themselves alive. It's so beautiful that you move there when you are sampling and you just touch one of these pine trees and you see feel all the aromatics that they release. Another protection strategy, because they produce those to avoid being eaten by insects, to avoid herbivory. We know that they are, as I mentioned, they keep their resources. They don't release much in the form of exudates, at least that's the idea, and they are short trees, okay? So we decided to study these terraces to analyze how microbial communities associate as soils degrade and as nutrients become limiting. The first thing that we did was to do a metagenomic analysis. And there are different keys here that I should mention before proceeding. When we did this metagenomic analysis, we actually went wild with it. The DNA extraction that we performed was on a large soil sample. We collected multiple samples from each terrace trying to follow an appropriate sampling strategy. We collected big amounts of soil, replicates, took 50 grams of that soil. We pulverized it under sterile conditions, liquid nitrogen sterile conditions, so all was homogenized. Then from that homogenate, we took 5 grams and we did our DNA extractions, DNA and RNA extractions. I'm only talking on DNA here. And this is going to become important, I think, for some of the results that you will see in a moment. So we did chemical analysis, metagenomes, amplicons, etc. I'm showing you just part of the results here. So our metagenomic analysis yielded millions of contigs, and we were able to actually assemble hundreds of prokaryotic MACs. What you see here is the distribution of the different contigs. Each little dot represents a contig there based on GC distribution and size. Sorry, you cannot see on that size. It is coverage here. Anything that you see in color is a contig for which we could assign some taxonomic level. But just look at this. All the gray that we have are things that we don't know anything about. So huge amount of data. From there, as I mentioned, we reconstructed MACs for prokaryotes, around 320 MACs, 5 archaeal MACs with a completion of 50 to 100% and less of 10% contamination. This will not tell you much, but what I want to show here are the different MACs in the y-axis, the 3 terraces, and you can see the distribution of microbial groups changing from terrace to terrace. And that was also seen by amplicon analysis and has been reported in these terraces, many other terraces as well. That's what I want to show here. So we have all these contigs, we have the MACs, and we wanted to know, is there greater prokaryotic genetic potential for the release and oxidation of ammonium in older terraces? Because we know that older terraces are limited by, well, there is no nitrogen for them. So we were thinking maybe prokaryotes are helping a lot up there, increasing the availability of nitrogen to their metabolic processes. Here, you can see the normalized coverage, a form of abundance of genes putatively coding for ureases, nitrogenases, and of course, ammonium monoxygenase. The urease is an enzyme that will take urea and will release 2 molecules of ammonia. So what you have here, or what you can see here, is that actually urease potential is higher in the young terrace. So we're like, damn, they are not the ones, potentially. Ammonia oxidation is potentially higher. Again, I'm not talking about function here because this is normalized abundance of genes, not transcription. The potential for it will be higher also in young terraces, in the young terrace. But these one look very interesting to us. Nitrogenases. The potential for nitrogen fixation seems to be higher in the older terrace. Nitrogen fixers, keeping the stability of those old terraces, all those beautiful plants growing. So maybe. But then we thought, well, who could be providing more of this nitrogen? There are studies, I will tell you more about that were done with measurements of the abundance of different forms of isotopically natural abundance nitrogen in these terraces, showing the potential paths for the distribution of nitrogen, who could be providing them. So what I want to say here is that the potential for nitrogen fixation was higher in the older terraces. And I forgot, did I mention the part in black up here? Maybe not, right? You can see there that we know that the mineral nitrogen pool goes from 300 milligrams of nitrogen per square meter in the young terrace to 0 in the older terraces. So that's why this, all that I was telling you here, is important. So again, who is facilitating this nitrogen mobilization or potentially doing it? There is another situation that I mentioned before, which is phosphorus availability is lower in older terraces. Previous studies have shown that total phosphorus decline from young to older terraces. Then there is phosphorus in older terraces, but up there, all the available phosphorus is occluded. That means is sequestered by minerals, aluminum, iron, and most of it is present in the form of organic phosphorus, phytate. So again, could prokaryotes be helping with the mobilization of phosphorus? We focus here on acid phosphatases, alkaline phosphatases, and also phytases for prokaryotes. We couldn't find significant differences for phytases, but you can see here that prokaryotic acid phosphatases, so the potential for their abundance, is actually higher in young terraces, which is pretty much the same situation as we were seeing with the nitrogen cycle or the genes that could favor nitrogen mobilization. The same prokaryotic potential for phosphorus mobilization is higher in the young teras. Well, we know for previous studies that in all or aging systems, fungi are super important. Here's a metagenomic analysis. So we decided to actually go on that route and to try to ask this question, could the key to the stability of all their terraces depend on fungi? Again, we have these contigs, and this took quite some time to me, but we established in the laboratory a cross-domain metagenome analysis pipeline so that we could actually retrieve information from these massive metagenomes from fungi, phages, all kinds of viruses, and also protists. With this, we were able to get max for all of these groups of organisms, which I am very happy about. Here again, you can see a distribution of some of these organisms. We have fungi, we have different kinds of viruses, bacteriophages, giant viruses. And again, the distribution is separated from side to side, which is something that you would expect. But the good thing here is that we retrieve 15 fungal max with a 50 to 70% completeness, 2 protist max, you can see the completeness there, and around 500 max, all with different, relatively decent quality. So, see my next one. I want to show you here. Fungal max from soil metagenomes point to a higher abundance of ericot mycorrhizal fungi in all terraces. And what you see in this plot will be the normalized coverage will be represented by the size of the circles of these max. You will see the different fungal max that we're able to assemble and their distributions among the terraces. And in this plot, but you can see the completeness here. In this plot, you can see the presence of absence for these different genes potentially coding for alkaline phosphatases, acid phosphatases, phytases, ureases, cellulases, silonases, lacases, and even polyphenol oxidases, indicating that this fungus or all these fungi have the potential to actually perform these tasks that you see there. So we are very happy with this result. Here we're supposed to mark the fungi that have been reported to be able to form these ericot mycorrhizal associations and the max for them. Well, super happy because at least by the time when we were trying to publish this paper, we didn't see any fungal max from a soil and from a metagenome reported. So that was a really cool thing. Now, if you ask me, my thinking is that our capacity to actually retrieve this information was due to our strategy to DNA extraction, the size of the sample that we were using, the strategy for DNA extraction that we were using, and also the depth of metagenome sequencing that we perform. Normally, when you do a metagenome sequencing, you go for 10 gigabase pairs, 20 if you're greedy, or 30 if you want to just use your money. Okay. In our case, we went for 30 in all of them. So it was a massive amount of data, a lot of data curation that I will tell you took years to get to this point back in the time when we were trying to set up all this. This is what I was mentioning before. There are nitrogen stable isotope analyses done at the ecological staircase that have indicated that ericoid and ectomycorrhizal fungi together with nitrogen-fixing bacteria are the key contributors to nitrogen supply in these nutrient-poor systems. And if you remember, I showed you also a higher potential for nitrogen fixation in these terraces, indicating, well, our results make sense in a way, But beyond that, I need to sell that again and again. We are able to get max, guys, for fungi, for protists. So we are very happy for that. So what are fungi potentially doing? What's the genetic metabolic potential of fungi in these retrogress ecosystems? Let me show you a big plot now. Again, we have normalized coverage, the 3 terraces, and the different genes that we were able to characterize. You can see there that there is higher potential for cellulose degradation in older terraces from fungi, higher potential for silanase degradation, lactases, and even polyphenol oxidases. So this is indicating potentially that fungi are high contributors of carbon cycling in the soil and the mobilization of carbon. For nitrogen cycling, higher potential of ureases in these older terraces, and for phosphorus cycling is the same story. Alkaline phosphatases, acid phosphatases, and even phytases. So they could be potentially contributing to the stabilization of these ecosystems in these degraded soils. And well, in 2017, Wardle in Boghurst published fungal-based energy channels are more dominant in retrogressive ecosystems. They did that by analyzing amplicon sequencing in these old terraces, and now we add a little bit more to this story by showing changes in fungal genetic potential in these terraces. Well, because you have a lot of data, you try to get everything you get out of it. And then you get to know really cool people that will teach you how to do network analysis. You have her here. So we decided to also do network analysis to analyze community connectivity and resilience or robustness. So we did amplicon sequencing for all potential markers that we had, and we created massive networks for each terrace. I want to show you the networks for the 3 terraces here, for the combined datasets. These networks contain the data for fungi, prokaryotes, and protists. What you see on the top is relative modularity, and you can see that relative modularity is increasing from young to old terrace, potentially indicating higher stability of these networks. And just look how the modules or the communities start to separate within them. And these will include organisms from all domains that we are analyzing. So to further analyze these networks, we were recommended to actually calculate robustness on the networks. And I was also very inspired by that paper of Maggie published in 2021. If you haven't read it, it's one of my Bibles, so just read it, I'll say. So we calculated robustness, which is the capacity of a network to transfer information when highly connected nodes are removed. Again, these networks are a theoretical reconstruction. What we could think is the structure of a community. So what you do here is that you go to these networks, remove these highly connected nodes, and measure the efficiency of the network to putatively move information along the nodes. And as you can see, robustness is higher in older terraces than in young terrace. Then we thought, well, here we have fungi, we have protists, we have prokaryotes in these networks. What will happen if we remove, for example, fungi from the networks, highly connected fungi? And then what would happen if we only remove highly connected protists with the robustness of the network? I have the results here. You can see what happened with the networks when you remove highly connected fungal nodes. Pretty much the older terraces have the lowest robustness, meaning potentially high need for the fungal communities to remain in these networks. And the same when you remove protists, lower robustness in the older network, indicating the potential need of protists in there. I need to say here, I have there a photo of a predatory protist. That's a ciliate. They were present in the older terraces, but I need to say that in these old terraces, a lot of the macroeukaryotic protist communities were microalgae, mixotrophic microalgae. So they are very important in these ecosystems. So what we have here is that in ecosystems, retrogression drives cross-domain microbial organization toward fewer but more interconnected lineages. And this organization is marked by increased integration of functional genetic potential, and well, that microbial networks adapt to maintain stability as soil age and fertility declines. That's what we are learning from this study. But again, I have always been, and I will say this, I have always been obsessed about understanding how things are connected. That's the view that I love about microbial communities. So observing these patterns across ecosystems will only get us so far. let me say, because I forgot. Now we're analyzing the metatranscriptomes also of these terraces. So what I show you here is just metagenome based. But our next question is, what do these interactions do to carbon cycling and community stability? That takes us to my next uh, project or idea, that some of you know about already, which is manipulating trophic complexity to understand its feedbacks to biological and environmental processes. We wanted to know what will happen if we can manipulate what you see in this image and how this will affect nutrient cycling, virulence, antimicrobial resistance, and more. But to get there, well, we should be able first to remove microbial groups. We have many papers published out there that are doing, for example, exclusion, beautiful work, or that do sieving of soils to separate fractions and then inoculating, I don't know, microcosms, mesocosms, etc. All super cool. But here we thought, how about we try to build this trophic complexity? The pieces, and we put them together. So for this, we needed to create first a multi-trophic enrichment. And this was work done by my undergraduate student, Matthew Heidenblut. You can see him there. He is coming back next fall to do his PhD in my lab. Excellent guy. So what we do with any environmental sample actually right now is that we will take a soil sample, we will incubate, give nice conditions. For example, these ones, 60% water holding capacity of the soil. Then we do gradient centrifugation. You cannot see really well here, but pretty much you will end up with a little bit of soil, a big density barrier, and above that, cells, active cells, that will include cells from all organisms. Then we take these cells and transfer them to these tissue culture flasks that will have soil extract or environmental extract sterilized, filter and filter and filter. So we're giving them the metabolites that they get in soil, the pH conditions. I'm not saying this is the same. We are, of course, selecting communities, right? And here we incubate, we observe, and we continue and continue. Let me show you here. You can see there how our enrichments look like, from different soils. You will see there ciliates, flagellates, you will see amoeba there. And well, we thought, okay, these are the cool things that we can see under the microscope. What's going on with everything else? So we did metagenomic analysis and applied our pipelines for metagenome characterization. From this enrichment that was prepared from a mixed conifer soil site, you can see here some of the communities present in the soil. These represent MACs. You can see also the pages that we were able to recover, and also, the MACs for some of the proteins present in these enrichments. So we were super happy with this. It's diverse. We have different groups, and we could potentially play with trophic complexity, separate its elements. To do that, in my laboratory, we use fluorescent activated cell sorting. You can see there our cell sorter. Really, it's a microfluidics-based cell sorting equipment inside of a PCR hood to try to keep everything clean. We take our enrichments, we pass them through that machine, and then we separate them based on size. These are the profiles of cell sorting. We have standards for size from 2 microns to 15. Here you can see when you add the whole enrichment, how it looks like, the distribution of sizes, if you mind that is overlaid with this. But what we do to separate the proteins is actually that we stain them with MitoTracker and LysoView to label mitochondria and also the lysosomes. And based on that, we can separate the population of proteins because we're using fluorescence activated cell sorting. And I want to show you here how they look like when they come out. This is very low magnification, but you can see the little guys just swimming around. Something that we love about this system, I won't try to, is that it's a very low pressure system because it's microfluidics based. Cells will go through and pretty much remain alive. We tested the sorting with other cell sorters, and usually our proteins just become cyst when they are going through because they are too stressed or burst open or who knows. But this one actually is giving us quite active communities. Is everybody doing well so far? Okay, I'll continue then. Well, now that we are able to produce these enrichments and sort them and separate communities, is when my PhD student Garrett Jenko started to do some work related to some other projects that we had in the past with some collaborators here, which is with his project, he's trying to understand how rhizosphere trophic complexity influences carbon and microbial dynamics. What I'm showing you here is pretty much the experimental setup. We start with 3 times autoclave soil. You autoclave soil for like 1 hour. You leave it resting at 30 Celsius for 2 to 3 days, leave things grow, autoclave again and autoclave again, just to deplete or remove some of the communities. You never remove everything. Then we introduce seedlings of Avena barbata that we surface sterilize. We grow them in sterile conditions, and then we pass them into these soils. And after that, Garrett wanted to create his own enrichment from a Mediterranean grassland soil in Hopland. Several of us work there or have worked there. And from there, he created these different communities that you see here. He added prokaryotes only, prokaryotes and protists, prokaryotes and AMF, spores of AMF, and everybody. Okay. For that, because we're inoculating things, we are worried about contamination, etc. We set up this plant room that you see there. We could control moisture. You can see here the humidifying system. They are closed. They have a light cycle, temperature. They were very careful to maintain the soil moisture in these and keep everything as clean as possible. They then leave the little seedlings established, and they come with a pipette and start adding the organisms really near to the plant, just trying to establish a community in there. From Garrett's enrichments, what did we add? I hope that you can read here. This is just a general plot showing what was present in this Hopland enrichment for Garrett. You have different organisms here in regards of prokaryotes. You will see several nitrogen-fixing bacteria that are growing in our enrichment in there. Mycobacterium, Clostridium, beautiful. We have the Lovibrio there. It's a predatory bacterium. It's just cool to see it. With regards of protists, these are the protists that are present in his enrichment. The first two, Spumelin and Sarcomonas, are flagellated protists. Then you will have others that are naked amoeba. You will have Procanthamoeba, Lobalfankia, and Canthamoeba, Philamoeba. And of course, we also have Bresaula there, which is a ciliate. So it's a very diverse community of protists. We took a bunch of soils. Our colleague Rina sent us a lot of soil. And then Nancy Johnson, she loves to work with AMF. She's from NAU, trained my master's students, Ivory, to actually extract spores from the soil. So we extracted the spores, cleaned them, and they focused mostly on Rhizophagus spores that they were getting from there. Well, now we inoculated, we let the plants grow, we went there to the different developmental stages of the plant. And over time, Garrett and a bunch of undergraduate, amazing, Jag, Will, and others, measured CO2 dynamics using this smart chamber that you see there. So we had the pods, we could actually see the smart chamber there, just put the system there and start measuring. So I want to show you the results here. As I mentioned, here you will see the time and time of sampling that will correspond to seedling, vegetative growth, flowering, desenescence. Actually, they flower and they senesce in the laboratory. And you can see our different treatments here. And I want you to pay attention to these two. The brown one is the plant that was placed in these 3 times autoclave soil. We didn't add anything to it. So there is the modeled trend. Here, in this dark green color, we have the bare soil because we added there as a control, maybe things will grow. And look at the linearity of it. Then, up here, you will have our low trophic complexity systems, which is where we just added the prokaryotes or where we added the protists and the prokaryotes. The beautiful thing, and maybe some of you will love that, is that when you have AMF, emissions are actually low. Maybe carbon is being redistributed through the soil. I mean, these are very, to me, cool signals, and I hope that you can appreciate them here. I'm sure you do. So now, Garrett is measuring carbon use efficiency of these soils. Oh, before I continue that, let me just show you here. So we also calculated the area under the curve of the plots that I showed you before to see the total CO2 emitted over the incubation time, every 4 days. And you can see here, when we have plant prokaryotes and AMF, the spread of the data, when we have everybody, the levels of CO2 that were emitted, and then the levels of CO2 when you just add certain groups in there without the AMF. And then, of course, you have the plant alone. Plant is also doing his thing, and bare soil there. Well, now Garrett is analyzing carbon use efficiency, as I mentioned. But because I was infected with the virus of the network, so I thought, well, let's do them again. Let's see what happened with them. Okay. So sorry about this one. I thought it would look better. But I want to show you here the networks for this plant alone or inoculated. As I mentioned, you will have organisms coming back to life there. It's not that you sterilize something with autoclaving and stays sterile, right? So in the network, you can see the little dots of all the communities that we could detect with, need to say, cDNA-based amplicon sequencing. We extracted RNA, we produced cDNA, we did amplicon sequencing, and then we created these networks, trying to avoid this problem that we have with relic DNA present in soils, etc. So you just get the active part. So you can see the networks there of the different stages. I'm not going to show you each of them because we tried that already, showing all the networks, and it doesn't go well. So let me try. Here, the little dots then now represent the background communities. You will see them now in this other network that corresponds to plant plus prokaryotes, AMF, and protein. You can see the little dots there, which are the organisms that we can find in the background. But anything that is in a larger dot are things that we added there. And we can see that maybe they remain, maybe they prevail. I don't want to say more of it. But you can see here the changes in the community. We can see that there are different organisms and that some of the things that we are adding there actually are active based on this analysis that we did. Again, robustness. This is where things to me get really interesting. You have robustness. I defined it before, right, with the terraces. How well can a network transfer information when you remove highly connected nodes? And look at this, when the plant is alone or uninoculated, it's kind of going up and down. But when you have microbes added, prokaryotes, you can see that it increases, and then decays. When you have AMF, you can see the same trend, but during senescence, the robustness is lower. Could this be an indication of a more loose association between plant and fungus because the plant is senescing? I don't know. Prokaryotes and protists. But then when we added everybody, you can see here that the change in robustness wasn't as marked. Maybe because they are all together, maybe because there is a high abundance of proteins that are releasing nitrogen and phosphorus to the system, even when the plant fungal association is becoming weaker. We don't know. We are doing metagenomes and metatranscriptomes and nutrient analysis to elucidate this. There is a lot of info. Okay, I promise I will go fast now because the next point is very important to me. So we can see here that high rhesosphere trophic complexity results in lower CO2 emissions, a difference that is more pronounced when AMF are present. Trophic manipulation alters bacterial network structure. Network robustness declines under plant senescence under prokaryotes and AMF. The presence of all microbial groups results in more stable networks even during plant senescence, and we're testing more of this. So here, again, fungi became the stars. That's why everybody loves fungi, right? But I'm like, we added protists there. What are they doing? Protists are often treated as background grazers. We wanted to know whether predation itself is an active driver of microbial function. So that's the next experiment. Here I want to tell you about this experiment in which we show that protease predation rapidly reshapes soy microbial gene expression linked to nutrient processing and environmental risk. This is work of my postdoc Dishant, again, Matthew. This was a simple experiment. We have three systems here. We have soil at 10% water holding capacity. We adjust the same soil at 60%. And then we have this soil adjusted at 60%, but we added a foreign community of proteins to see what will happen there. We did DNA and RNA, we did extraction, we did metagenomics and metatranscriptomic analysis. And the first thing that we wanted to know was, we know that if you add water to soil, you will activate proteins. I hope that everybody knows that. You just added a bunch of water and you're supposed to see them grow. But there was a lot of like people not believing, okay, when we were doing this experiment, like, let us show. So I want to show you here, this is a volcano plot showing only significantly expressed ribosomal markers for fungi, nematode proteins, and even tardigrades. On this side, you can see the groups that were significantly high expressed in the soil at 10%, water holding capacity, and in this one, those in which we added water. And I want you to see here that we have fungi, many of them. We have some nematodes. Most of them were plant parasitic nematodes, thankfully. We have even a tardigrade and some protists. But as you add water, you can see here the activation of all the protist groups being significantly more expressed than what is present in the soil. And here you have, ciliates, you have naked amoeba. There was even testate amoeba in there and amoeba and others. I'll tell you something. So I was showing there to my collaborators, like, well, but you have the means. You could count if they actually increase in density. Like, okay. So again, the same experiment. We did cell sorting, as I mentioned to you before, labeled the proteins, extracted the proteins directly from the soil, labeled them, use facts, and you can see there. When we have our soils at around 10% water holding capacity, we were able to count around 6,000 events for protists. As you add water, we got around 23,000. So there's a significant increase in protist density, just adding water. They were like, well, this study is about predation. What's going on with nematodes? Nematodes are important, right? And I'm like, well, we detected nematodes in the transcriptome that were plant pathogenic, most of them, plant parasitic, but still we will count them. So we counted them. You see here the water addition. We have 0.8 nematodes per gram of soil around that. It's presented just this for the sake of for you to see it. There is something that I need to show here. These experiments were done or incubated only 3 days. So imagine in only 3 days, you have this burst of protists. Now, also our jars were only 5 grams of soil. So the chances for us to add a predatory nematode or even a nematode were quite low. So we're like, well, this is about predation. As I mentioned before, we also added protists. We added in one of the treatments around 40,000 protists. These soils were from a mixed conifer soil site. We added protists from Hopland, California. They haven't seen the communities in these soils. They haven't seen each other. So you can see the organisms that we added there from this enrichment: Paramecium, Hilodonella, Cryptodiphugia, Paracircomonas, and so on and so on. All of them predatory protists. But will they survive reintroduction in soil? These enrichments that I show you, in liquid conditions have been running for 3 years. So we have been getting there. And somebody asked, when you will try to publish this, they will ask you, did they survive? Yeah, you're right. So to show that they survive reintroduction, we use QSIP, 18-0-based QSIP. So we had the same jars incubated with 16-0 or 18-0. We added the protists, again, 16-0, 18-0, extracted total DNA, did ultracentrifugation, separated the fractions, light DNA, heavy DNA for those organisms that incorporated the stable isotope 18-0, and we did sequencing. I want to show you the results. We can see here what is actively incorporating 18O when we just add water, what is actively incorporating 18O when we add our protist in water. And in these light blue colors, you can see the organisms that we added from our enrichment. And you can see that they are not present in the soil that we were using for the analysis. So we can say they are alive and they are potentially affecting the communities in the soil. Well, let's step back for a second. What happens with prokaryotic communities when you add water to a soil and you increase naturally occurring protist communities? In this plot is for differential expression. You see bacterial groups and the log-to-fold change from our metatranscriptomes. Right? Anything above this line, 0, is what is active when we added water, and anything behind here is what is active when you just have the soil. I want you to see some of the significantly ribosomally active groups when we just activated the natural proteins in the soil. Most of them are gram-positive organisms, or all of them. And if you can see gram-negative, go down in expression. So now, remember that I told you we added proteins that have never seen this community. What is going to happen? And we added them in excess. We added a bunch of them. I want to show you here. The things flipped. In this case, the less active communities, based on expression, were gram-positive. And the ones that apparently were surviving were all these gram-negative organisms. Later, because I'm not going to show you it here, I'm on time already, we did MAC reconstruction, and we could see that several of these groups actually were actively producing antimicrobial resistant genes, or expressing, not producing, antimicrobial resistant genes, secretion factors, virulence factors, potentially defending themselves. One thing that we couldn't detect a lot was in the gram-positive, several of these genetic elements that I'm telling you about. So we can see here that we added a community that has never seen the protist or vice versa. Bacteria haven't seen each other. They adapted, survived, and the things flipped in a way. At least that's what we can say now. Well, we are a soil lab in any way. As I told you before, when protists eat bacteria, they release ammonia, right? Ammonia. Another way in soils for the release of mineral nitrogen is through urea and the activity of ureases. And then you can see the whole nitrogen cycling. So we are thinking, are protists influencing nitrogen cycling? Here I want to show you genes involved in ammonia oxidation from archaea and from bacteria. You can see this is expression, and you cannot see well thing, but when protists were added, the expression of this MOA gene was significantly higher. So, because we added more protists, maybe there is more of the substrate, and more of these organisms are becoming actively oxidizing this nitrogen. What happens with the rest of the cycle? I won't get to it, all of it in detail, because, and I apologize, I shouldn't have done this. But you can see that in all cases, for nitrification and denitrification, the presence of protists actually increase the expression of genes involved in these steps, not for nitrogen fixation and NIPH. Also, we see that when protists are present, there is an increase in the expression of phytases, potentially releasing phosphate from phytates, from organic matter. And the same for some acid phosphatases. So protists are also changing community structure, but also influencing nitrogen and phosphorus cycling. I'm almost there, guys. What happens when a prokaryote faces a big predator? They can do different things. They can form a biofilm to protect themselves. They can run, flagellar activation. They can produce antibiotic and toxins trying to kill the protist, or they can induce secretion systems. When they produce antibiotics and toxins, other members of the community, to survive the exposure to these, will express antimicrobial resistant genes. I think that makes sense, right? So I want to show you the diversity of expressed antimicrobial resistant genes significantly increase when proteins are present. And you can see here the different genes involved in different mechanisms for antimicrobial resistance that are more highly expressed when proteins are present. So proteins may be a mean to increase antimicrobial resistance in ecosystems, right? Even if you haven't added an antibiotics to an ecosystem, there hasn't been anthropogenic, inputs. These soils were from a natural reservation, so there was nothing going on there, and still we can see these. Here, virulence factors. You can see that there is an increase in the diversity of expressed virulence factors when proteins are present. They are fighting, they are trying to survive prokaryotes, the presence of the proteins. And I want to show you really quickly that heat map or soil water proteins. So I didn't do this before. The different genes and the potential mechanisms in which these virulence factors act. In vitro experiments have shown that proteins can also facilitate horizontal gene transfer. When a bacterium is eaten, it goes into a vacuole. Maybe there is somebody already there. They are in other prokaryotes. They are in so tight proximity that you have an increase in gene transfer between them. This has been proven in vitro. In soil, we can see that there's an increase in the diversity of mobile genetic elements when you add water, but when you add proteins as well. When you activate proteins, and then when we added them, we can see an increase there. And this is another volcano plot just showing, that there is higher expression of these mobile genetic elements when you compare the protease treatment versus the dry treatment or versus the water treatment. So we can say that this is happening and proteins are mediating these processes. Dishant has this, my postdoc has this paper on the review now. And well, I'll take you to this summary. You saw it all, but predation takes proteins will go against a community, will homogenize it, and then will increase environmental risk, but also influence biochemical cycling. With this slide, I just want to close the presentation. Taking together, I hope I showed you that multitrophic interactions are not just a feature of microbial communities. They are a mechanism that links biodiversity, stability, and biochemical function across scales. And with this, I want to say thank you to all our collaborators. And here's a picture of my laboratory at NAU. I want to thank you for your attention, and I hope we have time for some questions.